A very good morning to my dear students of class 11 science. Welcome to biology class. So today we will begin with a new chapter, cell cycle and cell division, chapter number 10. Okay, so we all know that each and every organism, though it may be unicellular or multicellular, they are made up of cells. So that's why you say that cell is the structural and functional unit of life. So each and every organism is made up of cell. And in fact, the growth of the living organisms takes place by the help of this cell, because each cells undergo division, okay, each cells undergoes multiplication. So the growth of living organisms depends on the growth and multiplication of the cell. Clear? In case of unicellular organism, that means those organisms which are made up of single cell, their growth and development simply takes place by the help of the single cell, where the single cell undergoes reproductions and produces two or more new individuals from the mother cells. But in case of multicellular organisms, that means those who are made up of more than one cells, like we human beings, we are multicellular. So in our case, the growth and development, okay, so in multicellular organisms, the growth and development occurs from a single cell, and that single cell is called the zygote. Clear? And we all know that zygote is formed by the fusion of male gamete and the female gamete. Clear? So which is also called fertilization. So during fertilization, the gametes will fuse with one another and lead to the formation of a single cell structure called the zygote. Now through that zygote, the development of multicellular organism will begin. Clear? So therefore, we have got mainly two types of cell division. One is called mitosis and the second one is called meiosis. Mitosis, it occurs in the body cells or in the somatic cells. You've got the word soma, S-O-M-A. Soma means body cells. Okay, so somatic cells undergo cell division by the help of mitosis, whereas the reproductive cells or the germ cells, which will lead to the formation of gametes, it undergoes meiosis cell division. Okay, so gamete formation takes place by the help of meiosis cell division. That's why gametes are haploid. Mitosis is called equational division because the number of chromosomes in the daughter cell and the parent cell remains the same. But in meiosis, the number of chromosomes in the daughter cells will be reduced to half. So therefore, the gametes will be haploid. Clear? So gamete formation undergoes meiosis cell division, whereas the somatic cells or the body cells undergoes mitosis cell division. So each and every organisms, they mainly follow this pattern of cell division, that is mitosis and the meiosis cell division, which also signifies that each and every organisms have got the common source of evolution. Okay, so actually what is the cell division now? So cell divisions can be defined as the process by which a mature cell divides and forms the two daughter cells, which resembles the parental cell in a number of characters. It says that cell division, where the cell will divide, where the maternal cell will divide to form the two daughter cell. And we have got two types of division over here, mitosis and the meiosis. Clear? So here in cell division, the mature mother cell will divide into two daughter cells, and that two daughter cells will have the characters which are similar to that of the parents. Okay, that is called cell division, where the parental cell is being divided to form the two daughter cells with the characters which are similar to that of their parents. But it will depend on the process or the types by which it is dividing. That is, either it is divided by mitosis or by meiosis. Okay, so here the number of chromosomes depends on what basis, on what type the cell are dividing. If it is mitosis, then the number of chromosomes will be the same in the parents and the daughter. But if it is meiosis, then the number of chromosomes in the daughter cell will be reduced to half. Alright, okay, now let's go to the next one, which is called the cell cycle. Now what do you mean cell cycle? So cell cycle means the sequence of events which a cell undergoes from the end of one division to the end of another division or the next division. Okay, so from the beginning of or from the end of one division to the end of another division, we will see a sequence of events which will be going on, which we will discuss afterwards. Okay, which we will learn afterwards. So that particular events which will going to proceed from the end of one cell division to the end of another cell division is called cell cycle. There are lots of events over there. Okay, we have got a sequential cycle of events which will be going on from the end of one cycle to the beginning or to the end of another cycle. So that particular event is called cell cycle. So what happens during cell cycle? During cell cycle, the cell will prepare itself to undergo the process of cell division. So let's discuss about the cell cycle. Okay, so cell cycles is mainly divided into two phases or into two stages. One is called the interphase or the I phase, and second one is called the M phase. So we have got interphase and the mitotic phase. So if you look in the figure number 10.1, you can see the phases G1 phase, S phase and the G2 phase. Can you see over here? G1, S phase, G2 phase. This particular phase, it marks the interphase. Clear? And after the interphase, the cell will enter into the next phase, which is called the mitotic phase or the M phase. This one, M phase or mitotic phase. So we have got this sequence of events which will be going on during this I phase and the M phase, and this particular event which will be occurring, which is known as the cell cycle. So let's see the activity of the cell during the interphase and the M phase. What type of changes does occur in the cell? How does the cell will prepare itself to undergo the process of cell division? Now we are going to discuss. So we learn that the cell cycle is divided into two phases. First one, we have got the interphase or the I phase, and the second one is called the mitotic phase or the M phase. So let us go with the interphase or the I phase. Okay, so interphase or the I phase is the interval period between the two successive cell division. Okay, so the time period or the interval between the two successive cell division is called the interphase. And during this interphase, the chromosome materials will remain in highly attenuated conditions, but it will prepare itself to go to the next phase. So what are those phases or those changes that occurs in the cell during this I phase? So let's see down. Though apparently inactive, the nucleus and the cytoplasm are metabolically very active. So during this interphase, the nucleus and the cytoplasm will be active metabolically. There will be synthesis and storing of all those substances which are essential for the cell division. So here the cell is preparing itself to enter into the mitosis phase, than to the cell division phase. Now since the cell is preparing itself to enter into the cell division stage, it is also called preparatory phase, where the cell is preparing itself to enter into the process of cell division. Clear? So therefore this interphase is divided into three distinct phases: G1 phase, S phase and the G2 phase. G1 phase is also called the first growth phase or the post-mitotic phase. Then it will be followed by the second phase, which is called the S phase, which is also called synthesis phase. Then we have got the third phase, which is called the G2 phase, or also called the second growth phase. So let us talk about the changes which occurs during the first growth phase or the G1 phase. So kindly note it down. Okay. So during G1 phase, there will be initiation of DNA replication. Look over there. So this is the interval period between mitosis and the initiation of DNA replication. DNA replication means where the parental DNA will replicate to form the two daughter DNA. The young daughter cells formed by mitosis grow in size during this period. So here the daughter cells which are produced during the mitosis will grow in size. Okay, due to the increase in the volume of cytoplasm and the number of cell organelles. So during this G1 phase, the cell will grow in size. How does they will grow in size? By increasing the volume of the cytoplasm and by increasing the number of cellular organelles. Okay, now next thing is that during the G1 phase, DNA content remain constant. Okay, so therefore the time interval between the mitosis and the initiation of DNA replication is called first growth phase, and during this first growth phase the cell will simply grow in size by the increasing the volume of cytoplasm and by the increasing the number of cellular organelles, and during the G1 phase the DNA content remain constant, the number of DNA remains the same. Okay, because the time interval between the period of mitosis and the period between the initiation of DNA replication. So DNA content remain constant during the first growth phase. Okay, then from the G1 phase the cell will enter into the next phase, which is called the G2 phase. So once the G1 phase is over, now cell will enter into the next phase called the synthesis phase. Now during the synthesis phase there will be a replication of DNA. So the most important feature of S phase is that it is characterized by the replication of DNA. So here the single parental DNA will replicate to form the two daughter DNA. Okay, so during S phase the replication of DNA occurs. Clear? And DNA molecule of each chromosome replicates by the synthesis of a new DNA molecule. Clear? Okay, now this most important one, look over here. At this stage, that means during the S phase, each chromosomes carry a duplicate set of genes. Each cell retains its original diploid to a number of chromosomes with duplicate set of genes. The S phase in most eukaryotic cells lasts for 6 to 8 hours. Once the S phase begins, the cell enters the point of no return and has to undergo mitosis. That means once the cell undergoes the S phase and once there will be replication of DNA, now the cell cannot come back to the G1 phase, because now it has to enter into the next phase, G2 phase, and from there it has to undergo mitosis. And here the cell will retain its duplicate number of genes, or the constant number of genes, that is the diploid number of chromosome 2n. Now once the S phase will be over, once the DNA undergoes replication, once it will retain its diploid set of chromosomes, now it will enter into the next phase, which is called the G2 phase, or the second growth phase. Now let's see what are the characters or the changes that occur during the G2 phase. Now during G2 phase there will be further growth of the cell. Okay, so during this phase cell prepare itself to go into the division phase. That means from the G2 phase the cell will grow in size, it will prepare itself to enter into the mitosis phase or into the cell division phase. Okay, so here the nuclear RNA, ribosomal RNA, messenger RNA are all synthesized, because we have got three types of RNA: ribosomal RNA, messenger RNA, and transfer RNA. Okay, so here this mRNA, rRNA, and nucleolar RNA all will be synthesized. Then proteins for ester formation, spindle fiber formation are also synthesized during the G2 phase. Okay, and mainly this phase usually lasts for about 2 to 5 hours. So certain proteins synthesized in the G2 phase causes condensation of chromatin into the chromosomes to initiate mitosis. Clear? So here we are saying that during G2 phase a cell contains double the amount of DNA present in the original diploid cell. So what you learn here, that during interphase the cell will prepare itself to undergo the process of cell division. So it is also called preparatory phase. Okay, so we divide into three phases: first growth phase, second growth phase, and the third growth phase. First growth phase or G1 phase is the time interval between the mitosis and the initiation of DNA replication, and during this phase the cell will grow in size by increasing the volume of its cytoplasm and by increasing the number of its cellular organelles. Here the DNA does not replicate, but from the G1 phase, when the cell goes to the S phase, the DNA undergoes replication. It will retain its diploid number of chromosomes. Then from the S phase the cell will enter into the G2 phase, where the further growth of cell occurs because it has to enter into the cell division phase. So here the proteins for ester formation, proteins for spindle fiber formation will be synthesized. rRNA, mRNA, nuclear RNA will also be synthesized. So here the cell is preparing itself to enter into the cell division phase. So now after the interphase will be over, once the cell will go from the G1 to S phase and from S to G2 phase, now from there the cell will go into the next phase, which is called the M phase or the mitotic phase. Okay, so mitotic phase or the M phase, this phase follows the interphase and represents the phases of cell division, and it mainly divided in two types, kerokinesis and cytokinesis. Now what do you know kerokinesis? You have learned in class 10 that kerokinesis means the division of nucleus, and it will be followed by the division of cytoplasm, which is called cytokinesis. So first there will be kerokinesis, where there will be division of nucleus. A single nucleus will divide into two daughter nucleus. Then it will be followed by the division of cytoplasm, where a single cell will divide into two daughter cell. Okay, so this M phase is divided into kerokinesis and cytokinesis. Now those cells which do not divide further, okay, and which do not proceed beyond the G1 phase, they will enter into the next phase, which is called the G0 phase. Only those cells which do not divide further and which do not proceed further from the G1 phase, they will enter into the G0 phase. Okay, so those cells which are entered the G0 phase will stop dividing. Okay, they will grow in size and they will undergo differentiation to perform a specific or a particular functions, and such cells are also called quiescent phase. Okay, such cells are said to be in which phase? Quiescent phase. And these cells are metabolically active but do not divide unless these get the signal to divide. Clear? Okay, so here we have finished the cell cycle. All right, so let's remember we have got two phases, interphase and the mitotic phase or the M phase. So we have got G1 phase, S phase and the G2 phase in interphase, and we have got the M phase or the mitotic phase, where we have got kerokinesis and cytokinesis. There's no need to go in details about the events or stages of M phase or the mitosis, that is prophase, metaphase, anaphase and telophase. Okay, simply now we'll move into the next one, which is called the cell divisions. And there are mainly two types of cell divisions. I have told you that one is called mitosis and the meiosis, and you have a third type called amitosis, which is direct mode of cell divisions. Clear? Mitosis means the equational cell division, where the number of chromosomes in the daughter cells and the parental cell remains the same, that you know. Okay, and it occurs in somatic cells or the body cells. And meiosis is called reductional division, because here the number of chromosomes in the daughter cells will be reduced to half, and it occurs in reproductive cells or the germ cells. So in our syllabus we have to only go with the meiosis cell division. Mitosis is divided into prophase, metaphase, anaphase and telophase, that we know. Clear? And there will be formation of only two daughter cells. Clear? But now we will check out the events or the stages under the meiosis cell division, which occurs in the reproductive cells or in the gametic cells, and which will result into the formation of 4 haploid daughter cells. So let's check it out the meiosis. Okay, so meiosis involves two sequential cycles of nuclear and cell division. So here we have got two sequential cycles of nuclear division and the cell division. That means we have got meiosis 1 and we have got meiosis 2 cell division. Okay, so meiosis divided into two types. Listen very carefully: meiosis 1 and meiosis 2. But it occurs only a single cycle of DNA replication. Clear? Note it down that meiosis involves two sequential cycles of nuclear and cell division, which is called meiosis 1 and meiosis 2. But it involves only a single cycle of DNA replication. Okay. Next thing is that meiosis 1 is initiated after the parental chromosomes have replicated. Note it down. Meiosis 1 is initiated only after the parental chromosomes have replicated to produce identical sister chromatid at the S phase. Then next one, note it down, meiosis involves pairing of homologous chromosomes, which we will discuss afterwards when we go through the different phases of meiosis. So meiosis involves pairing of homologous chromosomes, homo means same, okay, and recombination between them. And the most important thing is that at the end of meiosis, 4 haploid cells will be produced. Listen very, very carefully. At the end of meiosis, 4 haploid cells will be produced, because we have got 2 types of meiosis cell division, meiosis 1 and meiosis 2. So during meiosis 1, 2 haploid cells will be produced. Now that 2 haploid cells undergoes meiosis 2. So from 2 haploid cells which are formed in meiosis 1, those will enter the meiosis 2 and will result in the formation of 4 haploid cells. So let's talk about the different phases or the different stages of meiosis cell division. So first of all, I've told you that we have got meiosis 1 and the meiosis 2. So kindly note it down. Okay, so let's talk about the meiosis 1, or the first meiotic division, which is also called heterotypic division, because here the number of chromosomes in the daughter cells will be reduced to half compared to that of the parents. So it is totally a reductional division. Like for example, human being, we have got 46 number of chromosomes, but during the time of meiosis cell division, for the gamete formation, the gamete that will be produced will be haploid. It will contain only 23 number of chromosomes. May it be sperm or the ovum, it will contain only haploid number of chromosomes, that is 23. It is called heterotypic division, okay, or also called reductional division. Now this meiosis 1 is divided into prophase 1, okay, metaphase 1, anaphase 1 and telophase 1. So first of all, let us talk about the prophase 1. Clear? So we are going to discuss about the first phase under the meiosis 1, that is the prophase 1. Okay, so prophase 1 of the first meiotic division is the longest phase. Clear? It's the longest duration, it's the longest phase of the first meiotic division, and also more complex one. Clear? So when we compare the prophase of mitosis and that of the meiosis 1, obviously the prophase of meiosis will be more complex and more longer than that of the mitosis prophase. So we can say that the prophase of the first meiotic division is typically longer and more complex when compared to the prophase of mitosis, and it has been further divided into the 5 phases. Now, what are those 5 substages or the 5 phases of prophase 1? They are mainly leptonema, okay, then zygonema, pachyonema, diplonema and dikinesis. We also call it leptoten, zygoten, pachygen, okay, diplotin and dikinesis. These are the 5 sub-stages or the 5 sub-phases under the prophase 1. Now let's see what are the events that occurs in each of these 5 sub-stages or the phases of prophase 1. So first one, we'll talk about the leptonema. So note it down. During leptonema or during leptotin, okay, the chromosome becomes gradually visible under the light. Note it down. During leptotin, the chromosome becomes gradually visible under the light microscope. The compaction of chromosomes continues throughout the leptotin, because chromosomes will be in the form of thread-like structures. So during leptotin, the compaction of chromosomes continue. Okay, so here we can say that during leptotin, the chromosome starts gradually to appear under the light microscopes, and the compaction of chromosomes, it will occur throughout the leptotin. Number 2 stage is zygonema or zygoten. So note it down. So during this stage, this is the most important one, okay, during zygoten or the zygonema, chromosomes start pairing together. So note it down. During zygoten, the chromosomes start pairing together, and this process of association is called synapsis. synapsis, synapsis. Even you can see in the point number B here also. Okay, so what you know is that during zygonema, the pairing of chromosome starts. Okay, so chromosomes starts pairing together, and that is called synapsis. And such paired chromosomes are called homologous chromosomes. Clear? So we said that the homologous chromosomes, the chromosomes which are same, okay, they will start to pair with each other. They will attract each other, and they will come close together, and they will pair up. and this pairing of homologous chromosome is known as synapsis. Now under the electron micrograph, it is seen that the chromosomes pairing or the pairing of homologous chromosomes, which is called synapsis, is accompanied by the formation of a complex which is called synaptonemal complex. So note it down, the next point number B or number 2, that electron micrograph of this stage indicates that chromosome synapsis is accompanied by the formation of a complex structure called synaptonemal complex. So here we can see the synaptonemal complex, the complex which will help in the pairing of homologous chromosome or which will favor synapsis. Clear? Next point, the complex which is formed by the pairing of homologous chromosomes is called a bivalent or a tetrad. So we look at the figure. Okay, so look over here, this is the interface one, and this we are talking about the leptoten, and we are moving to the next one, is called the zygoten. During leptoten, the chromosome starts to appear under the light microscope and the compaction of the chromosome continues, and from the leptoten the cells moves into the zygoten where there will be pairing of homologous chromosomes, and it's seen that this pairing of homologous chromosomes or the synapsis is accompanied by the formation of a complex called synaptonemal complex, and the synapsed homologous chromosomes will appear in the form of a bivalent or a tetrad. That means there will be 4 daughter chromatids. That's what it's called, tetrad. Okay. However, which are more clearly visible at the next stage. That means the tetrad or the bivalent will be more visible in the next stage, which is called the pachyten. Okay, so when we move from the zygoten to the pachyten, the tetrad or the bivalent will be more clearly visible. So pairing of homologous chromosomes occurs during the zygoten or the zygonema, which is the most important features of this stage. So from zygoten the cell moves into the pachytens where the bivalent or the tetrad will be more clearly visible. So note it down, pachyten, during this stage the bivalent chromosomes now clearly appears as tetrads. Tetrads means having 4 chromatids. Clear? So you can see over here, bivalents with 4 chromatids. Since there are 4 chromatids, it's also called tetrads. So during this stage, bivalent chromosomes now clearly appear as tetrads. Number 2, pachyten stage is characterized by, write down, it is characterized by the appearance of recombination nodules. Okay, so pachyten is mainly initiated or is mainly characterized by the appearance of recombination nodules. So let's check it out. Then note down the site at which the crossing over occurs between the non-sister chromatids of the homologous chromosomes. Now where does this crossing over occur? It said that crossing over occurs between the non-sister chromatids and is mainly seen by the appearance of recombination nodules. So here you can see the tetrad, bivalent with 4 tetrads with 4 chromatids. Now these 2 particular chromatids, they are not homologous, they are non-sister chromatids. But here you can see this particular point where there will be exchange of genetic material. So here you can see the formation of recombinant nodules where the crossing over occurs. Okay, so this is the most important features of the pachyten stage. So this stage is characterized by the appearance of recombination nodules, the site at which crossing over occurs between the non-sister chromatids of the homologous chromosomes. So crossing over means the exchange of genetic material between the 2 homologous chromosomes. Clear? So at this particular point where there is mainly crossing over between non-sister chromatids of the homologous chromosomes, there will be exchange of genetic material. And crossing over is also an enzyme mediated process. That means For the exchange of genetic materials, we need the help of an enzyme. So crossing over is an enzyme mediated process, and the enzyme that will help in crossing over is called recombinase. Write it down: RECOMBINASE. So crossing over is an enzyme mediated process, and the enzyme is called recombinase. RECOMBINASE. And what is crossing over? Crossing over is the exchange of genetic material between the two homologous chromosomes. Okay, and crossing over leads to recombination of genetic material on the two chromosomes, or the formation of genetic combinates. Clear? And the recombination between homologous chromosomes is completed by the end of Pachytene, leaving the chromosomes linked at the site of crossing over. So we are moving to the next phase now. From the Pachytene, we are moving into the next phase, which is called the Diploten. Okay, so from Pachytene, the cell will move to the next one, which is called the Diploten. Now let's see what are the characteristic features of the Diploten phase of the cell divisions. So first, write down: the beginning of Diploten is recognized by the dissolution of the synaptonemal complex, the complex which was formed in which phase? In the zygotine phase. Okay, we have learned that mainly during the zygotine there will be formation of synaptonemal complex. Okay, so during this zygotine phase there was formation of synaptonemal complex. Now in case of this Diploten phase, that synaptonemal complex starts to dissolve. So this phase is recognized by the dissolution of the synaptonemal complex which was formed in the zygoten or in the zygonema. Okay, and it is also marked by the tendency of the recombined homologous chromosomes of the bivalent to separate. Okay, homologous chromosomes has been paired during which phase? In the Pachytene phase. But in Diploten phase, now those homologous chromosomes of the bivalent starts to separate from each other, except at the site of crossover. Except the site of crossovers, that means crossing over sites, the homologous chromosomes, they will start to separate from each other. Okay, and they will form a egg-shaped structure which is called the chiasmata. Now I'll show in the diagram which one is chiasmata. If you look over here in Diploten phase, okay, it says that the homologous chromosomes except at the site of crossing over. So this one is the point of crossing over. If you remember, this is the site of crossing over. Okay, this is the point of crossing over. Except at the point of crossing over, the bivalent chromosomes, they will try to separate from each other. Okay, they have the tendency to separate from each other, and they will form a egg-shaped structure. They will form a egg-shaped structure. Now this egg-shaped structure is called chiasmata. CHIASMATA. So here you can see egg-shaped structure has been created, and this structure is now known as chiasmata. Now from here, from Diploten, the cell will move into the next phase, which is called the dikinesis. And in dikinesis, we can say that it is the final stage of prophase 1, and it is marked by the end of chiasmata, or we can say the terminalization of chiasmata. And during this phase, the chromosomes are fully condensed, and the meiotic spindle is assembled to prepare the homologous chromosomes to separate. So one more time I'm saying that during dikinesis, there will be terminalization of chiasmata, and there will be formation of meiotic spindle fiber. You can see the spindle fiber. Okay, there will be formation of spindle fiber, so that it will be easier, or it will prepare the homologous chromosomes to separate. And by the end of dikinesis, the nucleolus disappear. The nuclear membrane or the nuclear envelope also breaks down. Here you can see the nuclear membrane, it was there, but once we are moving from the leptoten to the zygoten to the packeten, diploten, the nuclear membrane slowly starts to disappear, and when it moves to dikinesis, it will totally disappear. So nucleolus will disappear, and the nuclear envelope or the nuclear membrane will also break down or disappear in dikinesis, and dikinesis represents the transition to the next phase, which is called the metaphase. So we have finished the prophase 1. Now from prophase 1, the cell will enter into the next phase, which is called the metaphase 1. So now what are the events that occurs during the metaphase 1? So note it down. The bivalent chromosomes, they will align on the equatorial plate. That means they will come to the center. Can you see over here that all the chromosomes, that is the bivalent chromosomes, they have aligned at the equatorial zone or at the center. So this is the most important phase of the metaphase, important feature of metaphase. Okay, so the bivalent chromosomes, they will align on the equatorial plate, they will come to the center. The microtubules from the opposite pole of the spindle fiber attach to the pair of homologous chromosomes. Now you can see that the microtubules from the opposite pole, this is the first pole and this is the opposite pole. So these are the two opposite poles, and these are the microtubules which are present at each respective poles. Now these two microtubules, they will pass from the, or they will attach to the pair of homologous chromosomes. Okay, so these microtubules, they will attach to the pair of homologous chromosomes during the metaphase. So during metaphase, the bivalent chromosomes, they will align on the equatorial plate, and the microtubules from the opposite pole, they will attach to the pair of homologous chromosomes. Then from metaphase 1, the cell will move to the next one, which is called the anaphase 1. So what are the changes or the events that occurs during the anaphase 1? So the most important one during anaphase 1 is that the homologous chromosomes start separating. Can you see over here? The homologous chromosomes, they start moving to each other. So here you can see that bivalent one, but during anaphase, what I mean is that they start separating. Can you see over here? They start separating and they are moving towards the opposite poles. So now the homologous chromosomes have started separating and started moving towards the opposite pole during the anaphase 1. But the sister chromatids, they remain associated at their centromere. Only the homologous chromosomes separate during anaphase 1, but the sister chromatids, they will remain associated at their centromere. So here you can see the sister chromatids which are being joined by the help of centromere. So this is the most important feature of anaphase 1. Now from the anaphase 1, the cell will move to the next phase, which is called the telophase 1. Important feature is that nuclear membrane, you can see over here, nuclear membrane reappears, and the nucleolus will also reappear during the telophase 1. Then cytokinesis follows, and this is also called as the diet of cells. So at the telophase 1, once these homologous chromosomes will reach their respective poles, there will be reformation of nuclear membrane, and there will be reappearance of nucleolus, and it will be followed by the division of cytoplasm, which is called cytokinesis, and there will be formation of two daughter cells, which are also called diet cells. So you can see the daughter cells. So from one mother cell, there will be formation of two daughter cells at the end of meiosis 1. Clear? Is it clear? So we have finished the meiosis 1. So at the end of meiosis 1, there will be formation of two daughter cells. Now these two daughter cells has to enter into the next one, which is called the meiosis 2. But there's a particular interval between the meiosis 1 and the meiosis 2. So that particular time period or the gap between meiosis 1 and the meiosis 2 is called interkinesis. So what is interkinesis? This is the interval between the first and the second meiotic division. So the time period or the gap between the first meiotic division and the second meiotic division, it's called the interkinesis. So after meiosis 1, we will find interkinesis, the time period or the gap. Then we'll move to the next one, which is called the meiosis 2. Okay, so meiosis 2 is also divided into prophase 2, metaphase 2, anaphase 2, and the telophase 2. It will have the same event that was occurring in meiosis 1, that is in case of prophase 1, metaphase 1, anaphase 1, and telophase 1. So we have got the same type of events here also. Okay, so let's see the the meiosis 2 of meiosis cell division. So meiosis 2, is essentially a mitotic division. That means here the number of chromosomes will remain the same. That means the two daughter chromosomes which was formed in the first meiotic division here, okay, it will be haploid because it is a heterotypic division. If the number of chromosomes, if it was 46 over here, then here in case of two daughter chromosomes it will be reduced to half. It will be 23 and it will be 23. Now these two daughter cells are entering into the second phase called meiosis II cell division. Here you can see. Okay, now these two cells are entering into the second phase called the meiosis II cell divisions. But the meiosis II cell division is simply a mitotic cell division. Okay, simply a mitotic cell division, because here the number of chromosomes in the daughter cells, in the two daughter cells, will remain the same. So we said that the second meiotic division is essentially a mitotic division and is separated into the same four phases or stages, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. So in prophase 2, the chromosomes, listen very carefully, in prophase 2, the chromosomes reappear, the nuclear membrane disappear and there is formation of spindle fiber. Okay, and each chromosome consists of two distinct chromatids attached to the single centromere, and this prophase 2 is initiated immediately after the cytokinesis. Clear? And usually before the chromosomes have become fully elongated. After prophase 2 we have got metaphase 2. Here also the chromosome will align at the equator. Okay, like in case of metaphase 1. Here also in metaphase 2, the chromosomes will align at the equator and the microtubule from the opposite pole will attach to the kinetochore of sister chromatids. Then it will be anaphase 2. Again there will be simultaneous splitting of the centromere of each chromosomes which are holding the two sister chromatids, and they will move toward the opposite poles. Now here the sister chromatids are moving toward the opposite poles. In anaphase 1, the homologous chromosomes was moving towards the opposite pole, whereas the sister chromatids was attached to their centromere. But in anaphase 2, now the sister chromatids will start moving towards the opposite pole. Okay, then we have got telophase 2, where the two groups of chromosomes once again get enclosed by a nuclear envelope which was missing in anaphase 2. Cytokinesis follows, resulting to the formation of tetrad of cell, that is 4 haploid daughter cells. Okay, so telophase 2 will end the process of meiosis cell division. So therefore after the end of meiosis 2, there will be formation of 4 haploid daughter cells. So second meiotic division you can see over here. So there will be formation of 4 haploid daughter cells at the end of meiosis cell division. But simply meiosis cell division will follow the process of mitotic cell division and same type or same stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear? So here you can see metaphase 2, chromosome coming at the center. Okay, microtubule passing through the kinetochore of sister chromatids. Then in case of anaphase, the sister chromatids are moving toward the opposite poles. You can see over here sister chromatids have been separated and they are moving toward the opposite poles. Okay, this is the first daughter cell, this is the second daughter cell. So it's like very carefully. Again, during telophase, they will reach to opposite poles, nuclear membrane reappear, nucleus reappear, it will be followed by cytokinesis, and there will be formation of 2 daughter cells. So from 1 daughter cell there will be formation of again 2 daughter cells. Again from here the second daughter cell there will be formation of 2 daughter cells. So that means from these 2 daughter cells, which are the product of first meiotic cell division, listen very carefully, these 2 daughter cells are the product of first meiotic cell division, and these 2 daughter cells have entered the second meiotic division. So from these 2 daughter cells there is formation of how many daughter cells? 4 haploid daughter cells. So at the end of meiosis there will be formation of 4 haploid daughter cells. So now what is the significance of meiosis, or what is the importance of meiosis cell division? We are going to discuss. First one, meiosis cell division will help in the formation of gametes. So meiosis produces haploid gametes for sexual reproductions, and the number of chromosomes will again be reserved or again will be conserved by the help of fertilization. Number 2, it will help in the maintenance of chromosome number and the stability of species. Okay, because in sexually reproducing organisms, it will help to restore the diploid number of chromosomes, and will maintain the stability of a species. By the help of meiosis, there will be the introduction of variation. There will be crossing over, where there will be exchange of genetic material between the two homologous chromosomes, and this particular crossing over or the exchange of genetic material between the two homologous chromosomes will help to introduce variation. So that's why there will be variation in the offspring produced by the mode of sexual reproduction. The number 4, it will help in the interrelationship between living organisms. So details of meiosis are essentially similar in all the sexually reproducing organisms, and they will show basic similarity and relationship among them. So this is the significance of meiosis. Now you will go with the table number 10.3, where we will just simply see the difference between mitosis and meiosis. You simply go through this table. Okay, now let's see the type of meiosis. We have got 3 types of meiosis. Gametic meiosis, which occurs in the organisms, in the diploid organisms, for the formation of gamete, and gametes are haploid. Okay, so we said that this type of meiosis occurs in organisms having diploidic life cycles, where gametes represent the haploid phase, which form the zygote, and the zygote forms the diploid phase. Number 2 is zygotic meiosis. This type of meiosis occurs in the organisms having haploidic life cycles, where zygote is only the diploid phase, okay, while the adult is the haploid phase. So here in zygotic meiosis, the diploid phase is only the zygote, whereas the adult is the haploid one. Okay, so in this case, the first zygote division is reduction division, and the adult and the gametes are found by the haploid. Then we have got sporogenetic meiosis, which occurs in case of plants, where gametes are produced by mitosis. So meiosis occurs at the time of spore formation only. So these are the 3 types of meiosis cell division. So here we have finished the chapter cell division and cell cycle. Kindly follow the new reduced syllabus. Okay, so we have finished this chapter cell cycle and cell division. For next class, we will begin with the new chapter. Thank you, and see you in next class.